6-PPD-quinone is a highly prevalent, rubber-derived environmental pollutant. As tires degrade on the road, they shed this chemical constantly, embedding it into our water systems, air, and soil. Its sheer ubiquity has prompted a critical scientific question. Does prolonged exposure to this modern synthetic compound trigger or accelerate the onset of Alzheimer's disease? Historically, establishing that kind of connection is notoriously difficult. Environmental toxicity claims usually rely on broad population studies, tracking associative trends rather than proving a direct molecular cause. Finding a true mechanistic link requires something much harder. It requires tracking the exact physical interaction between a microscopic pollutant molecule and the specific regulatory proteins inside the human brain. Isolating those exact protein interactions against the massive noise of human biology requires moving beyond traditional laboratory trial and error into the realm of advanced computational toxicology. This schematic details the study's methodology, a rigorous 6-step in silico pipeline designed to filter thousands of biological variables down to specific targets. The researchers started by mapping the predicted targets of 6-PPD-Q directly against known genetic databases for Alzheimer's disease. From there, the pipeline systematically layers network topology, transcriptomics, genetic susceptibility, machine learning, and finally 3D structural biology. This multi-omics funnel bypasses the limitations of animal models, allowing scientists to test for neurotoxic mechanisms directly against human data. By the end of these 6 steps, we can isolate the exact protein networks that 6-PPD-Q hijacks to drive neurodegeneration. This Venn diagram illustrates the initial mathematical intersection. Cross-referencing 6-PPD-Q's predicted targets against Alzheimer's genetic databases yields exactly 92 overlapping genes. However, a list of 92 independent genes is too broad to explain a specific toxicological mechanism. We must determine how these genes interact with each other in a coordinated network. Researchers applied a protein-protein interaction topological analysis to map this network, calculating mathematical centrality to find the critical structural hubs. That analysis identified 3 specific genes ranking at the absolute top of the network: NFKB1, GSK3B, and PIK3CA. But mathematical interaction on a server does not guarantee biological relevance. We have to prove these genes are actually active in the specific areas of the brain destroyed by Alzheimer's. This FUMA tissue expression chart tests for that exact physical presence. The dense cluster of red bars indicates severe expression enrichment in specific anatomical zones. The data shows massive expression specifically localized in the brain cortex, amygdala, and basal ganglia. This localized enrichment confirms these overlapping genes are not statistical noise. They sit precisely in the disease's blast radius. In step 3, the pipeline tests whether these specific targets behave differently in diseased brains compared to healthy ones. To find out, researchers pulled bulk RNA sequencing data derived directly from post-mortem human temporal lobe samples. These plots visualize the differential expression analysis. Panel A shows clear statistical separation between Alzheimer's patients and control groups, while Panel B tracks how the behavior of our core hub genes actively diverges depending on the disease state. Because RNA data only confirms that the genetic instructions are altered, scientists sought physical proof of the resulting proteins. Panel F provides that proof. This immunohistochemical staining confirms a heavy physical presence of key targets, specifically NFKB1 and GSK3B, inside actual human cerebral cortex tissue. Step 4 transitions into Mendelian randomization to evaluate the genetic backdrop of these targets. In neurology, the ApoE gene cluster is universally recognized as the strongest genetic risk factor for late-onset Alzheimer's. These charts map our study's core genes directly against the architecture of the ApoE and ApoC1 loci. The towering peaks of the GWAS and pQTL data align perfectly with the genetic regions below. These intense protein-level associations reveal a highly volatile genetic environment, perfectly primed to react to external stressors. Steps 3 and 4 ground the computational model in biological reality. The targets exist in the correct brain regions, their behavior is demonstrably altered in patients, and they operate against a backdrop of severe genetic vulnerability. Validating the targets is necessary, but it does not tell us which genes actively drive the disease and which are merely reacting to the damage. Step 5 addresses this using machine learning. Researchers trained an XGBoost classifier, an algorithm designed to predict whether a brain sample has Alzheimer's based exclusively on the expression levels of these core genes. To understand how the algorithm makes its decisions, researchers apply SHAP values. This mathematical technique cracks open the model to measure exactly how much weight it assigned to each individual gene. This SHAP summary plot ranks those variables. 5 genes overwhelmingly contribute to the model's accuracy: PTGS2, KIT, PIK3CA, NFE2L2, and NFKB1. SHAP values also track directional impact, measuring the degree to which a gene's expression actively pushes the algorithm toward a specific diagnosis. Panel B isolates a representative control sample. You can see the competing genetic forces at play, with blue blocks pushing the final prediction left toward control, while red blocks push right toward Alzheimer's. The data shows that high expression of PTGS2 and NFE2L2 acts protectively. They anchor the model in a healthy status. Conversely, high expression of KIT, PIK3CA, and NFKB1 acts destructively, overriding those protective signals and driving the model toward an Alzheimer's classification. In human biology, these specific genes are the primary regulators of inflammation and oxidative stress in the brain. By quantifying their impact, the machine learning step isolates the precise molecular levers that, if disrupted by a pollutant, would tip a healthy brain into neurodegeneration. A statistical prediction fails if the 6PPD-Q molecule cannot physically bind to these specific protein levers. Step 6 tests this through molecular docking. This in silico structural biology process simulates the thermodynamic fit between the 6PPD-Q molecule and the exact 3D geometry of our top machine learning targets. These structural diagrams visualize the docking results, confirming strong predicted binding affinities for targets isolated by the machine learning model. The simulation calculates highly stable structural connections. PTGS2 shows a binding energy of -8.4 kilocalories per mole, and NFE2L2 returns -8.3. The specific orientation of these chemical bonds suggests that 6PPD-Q physically blocks the normal enzymatic functions of these proteins. Furthermore, NFE2L2 and GSK3B both demonstrate amplified regulatory effects specifically under Alzheimer's conditions, linking their structural vulnerability directly to the disease state. These physical docking models provide the final proof of concept. The abstract statistical association is now a documented biomechanical threat. The pipeline uncovers a highly specific biological sequence. When 6PPD-Q crosses into the brain, it bypasses random tissue damage and slots directly into key regulatory nodes. This physical binding triggers a 3-pronged mechanical failure: the induction of severe oxidative stress, the activation of a runaway neuroinflammatory cascade driven by NFKB1, and the targeted disruption of kinase signaling networks. Together, these 3 dysfunctions form the exact biological precursors to the widespread synaptic failure observed in Alzheimer's pathogenesis. By stacking transcriptomics, machine learning, and molecular physics, researchers can now map the precise pathways exploited by environmental toxins. 6PPD-Q represents a mapped biochemical trigger that exploits the exact structural and genetic vulnerabilities of the aging human brain.